Welcome on this Rare Disease Day to the first DNA Dialogue of 2024. In the spirit of reconciliation, now more than ever, I acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. Today, I'm joining you from the lands of the Bunurong people of the Kulin Nation. I pay my respects to their elders, past and present, and extend that respect to all First Nations peoples today. So DNA Dialogue, as you are probably well aware by now, is a forum that we bring people together to share the genomic innovations from around the world with really leading experts sharing their work. You're welcome to ask questions during the seminar today using the Q&A function, and I'll get to as many of those as I can at the end of the presentation. So I'm very pleased today to be joined by Mashaal Sohail. She is an Associate Professor at the Center of Genomic Sciences of the National Autonomous University of Mexico. She completed her PhD in systems biology at Harvard and undertook postdocs at the University of Chicago and at Mexico's National Laboratory of Genomics for Biodiversity. Her lab focuses on population and complex trait genomics with an interest in underserved groups from Mexico and Pakistan. Today, Mashaal will be speaking to us, sharing a presentation entitled Mexican Biobank Advances Population and Medical Genomics of Diverse Ancestries. Now, Latin America continues to be severely underrepresented in genomics research, and fine-scale genetic histories and complex trait architectures remain hidden owing to insufficient data. This is a topic that is very current and topical for us in Australia as well, of course. To fill this gap, the Mexican Biobank project genotyped over 6,000 people from across Mexico, creating a valuable nationwide GenFEN database. And the study provides insights into the genetic histories of individuals in Mexico and dissects their complex trait architectures, both of which are crucial for making precision and preventative medicine accessible worldwide.
Okay, well, hi everyone. It's a great pleasure to be here. So today I'll be speaking about the Mexican Biobank and how it's advanced population and medical genomics of diverse ancestries. This project started basically as a collaboration between Langhebius Investap and the National Institute of Public Health in Mexico. And the idea was to genotype the samples that had been collected by the National Institute of Public Health as part of their year 2000 survey across the country, and to genotype these samples and to use them for a number of complex trait and population genetic analysis like the ones that I'll be talking about today. And so this project was funded by a joint call between the Newton Fund and the CONACYT in Mexico. There's a larger collaborative network that includes University of Oxford, Chicago, and Leicester. So what's nice about this project and where it's really a first is that it covers in Latin America, that it covers the diversity across the country. So we have all 32 states covered, and we're enriching for rural localities. So 70% of the biobank is from rural localities. And so in this first phase of the project that I'll be showing some results for today, we have 6,000 individuals genotyped on the ENSA. And all the genotyping was done in Mexico. So the genotyping was done here. And to give a face to a name, this is Langhebius Investap, where I started as a postdoc on this project now about 5 years ago. And so where all the genotyping was done, where we led all the analysis, despite having this larger international network. And this is the Center for Genomic Sciences of the UNAM, where I then took a faculty position about over 3 years ago now, and where I have my own group working with the Mexican Biobank, but also other projects in South Asia and so on. So this project, the flagship paper recently came out just last August, and it, along with the study, another study also came out, which is on the Mexico City Prospective study, which is another biobank that came out of Mexico. And while it has about 150,000 individuals, that other biobank is restricted to Mexico City. So the two data sets together really, I think, promise a very bright future for genomics in Mexico, and a lot of discoveries to be made, and together they were given the cover of Nature for Mexican Genomics here, showing indigenous art of the Villarrica or Huichol people. So this project basically came at a time when there's a persistent bias in genome-wide association studies, and while it continues to improve, still remains quite striking. And I think a question to keep in mind sort of throughout this talk, and that we sort of tackled throughout this first project and studies, why we should diversify genetic phenotypic databases. Because this question has a difficult history and likely a difficult answer, but we can try to unravel some of the facets here. So to think about that, we need to first think about the concept of genetic ancestries. This is something that's been conceptualized and inferred using methods such as principal components analysis and admixture. And it's really a concept to capture the way in which we vary genetically from each other due to our near and distant relationships. And the best way to think about it really is in terms of a tree, something like this one, which is taken from this working group that I'm part of, from the Safra Center for Ethics, to think about the normative implications of use of the words population and ancestry in genetics research. And here the idea to really keep in mind is ancestry is really sort of a continuous and multidimensional concept. And anytime we're sort of creating categories with it, we're doing a discretization of what is actually a continuous thing. And in that sense, we're also always, when we discretize, sort of focusing on a particular time slice and what is really like a large tree, sort of showing all the ways in which we're connected. So keeping this in mind throughout the talk. So first, what we did was we used principal components to look at the Mexican Biobank samples. So here they're shown in gray with respect to various individuals from the 1,000 genomes and HGDP data sets. And so you can see they mainly fall, unsurprisingly, along a climb between indigenous individuals here sampled from Oaxaca, the Benza from a previous study, the NMDP, and basically Eurasian individuals here. And you can start to see also a pull towards African individuals here and a slighter pull towards Asian individuals here, which I'll talk a bit about shortly. So if we do the same thing of dimensionality reduction on just the individuals of the Mexican Biobank now without any references. So to do this, we think of another time scale, right? And we worked with our collaborators in anthropology to regionalize Mexico into these regions that are really defined by cultural context and civilizational history. So here we have the north of Mexico, the north of Mesoamerica, the occident of Mexico, the Gulf of Mexico, the center, Oaxaca region, and the Mayan region. And so when we do it like this, and we color the individuals by the region that they're from, what we can see is, in particular, the Mayan region sort of standing out a bit more in both the PCA and the UMAP analysis, while still, as you can see, with the overlap, sharing some ancestries with the Gulf and center of Mexico, and this will come up again. Whereas with the rest of the individuals we see, it's clear that there is a gradient, but that there is a lot of sort of overlapping and so on. And I think that reflects all the migrational sort of histories between the regions up to year 2000. So this can be older relationships between these regions, which are evident from oral histories, as well as, of course, more recent movements in the current era. So we can use also another approach, also commonly used, called admixture, again, with reference individuals from all over the world to look at, sort of try to define, quantify these ancestries. And if you do this at k=5, which is mainly capturing these sort of continental ancestries in some sense, thinking back to that tree I just showed. So what we can see is that in red, we have these ancestries that are predominantly found in indigenous individuals, and these, as you can see, vary. So here, these are placed sort of from north to southeast, the states of Mexico. And so the indigenous ancestries are increasing as we move towards the south and southeast, and ancestries that are commonly found in Europe, as well as in the Middle East and South Asia, are increasing as we move towards the north of the country, so in contrast. And so here, this is just for visualization, pulling these out. And maybe you can notice that we do have African ancestries in blue in all the states, as well as small, but definitely significant amount in purple here of ancestries from East Asia, in particular in the state of Guerrero. So I won't have too much time to talk about this today, but this goes back into a very interesting history of the Manila galleon trade from the Philippines to Acapulco, starting in the 16th century when both were Spanish colonies, and that has left its mark in Mexico today as well. And there's a paper fully dedicated to this topic, which is Juan Esteban Rodriguez et al. So of course, as I mentioned, like we can do admixture for a range of Ks, and this is really capturing again sort of those different time slices and resolutions that we should think about ancestry. And so I had talked about K equals 5, but as we do this at higher Ks, we start to see, I think the major thing that comes up is the breaking down of ancestries within what we may call indigenous ancestries, and we start to see sort of more resolution within that, as well as really within any ancestry, such as what we may call European and so on. So just in terms of a sort of very quick picture summary, these are the main ancestral sources, if we had to put them into these buckets, found in Mexico, in the different states. And indigenous ancestries are the predominant ones in the Mexican Biobank for almost every state except 3. We see, about around roughly 5% African ancestries and a small amount of these East Asian ones, the highest in Guerrero. So sort of diving deeper into making, the ancestry profiles of the Mexican Biobank, we use this approach of what has been called the archetypal analysis, which also tries to model based on source populations, but it's a bit different from admixture because it sort of, I think, is better at highlighting the continuous nature of the population structure. And so when we apply this approach to just the Mexican Biobank individuals here, shown first on their own, colored by the different regions. So again, we can do this sort of assuming 3 source populations and up to 10. You can see, I think the thing to sort of note is just the diversity that is present within just this data set because of the sampling approach that we took, sampling from each and every region. Just the Mayan region, for example, appears with this modeling approach to be coming from several different sources. And if we redo this analysis now, including these reference individuals from the 1,000 genomes in HGDP, so here shown in color, while the MEXP are shown in gray on the left, and on the right, the references are shown in gray and the MEXP in colors. So again, I think it just sort of highlights how much of an effect sampling has in what we perceive about diversity. So in this particular image, for example, because we have very few samples from Africa, it's just from the 1,000 genomes, all of Africa is modeled to coming from one source in the presence of all these samples. And given the amount that we have from the MEXP, you can see the Mayan individuals being shown to be modeled from several different sources. And again, standing out just like in the PCA and UMAP compared to the other regions that tend to have more overlap. So this is all in terms of thinking more globally about ancestries. Another thing that we can do, and that has commonly been done in individuals from Latin America, is to use an approach such as RF-mix or PCA-ADMIX to infer local ancestries along the genome. And so you can color the genome with respect to sort of the origins of those segments of the genome. And then the window there is that you can use only those segments of the genome from a certain origin, and that can allow you to ask more interesting kinds of questions. So what we did was we used Genomics, which is the newest approach to call local ancestry, and colored our MEXP individual genomes by these 3 different origins. And then we worked with only the segments with indigenous ancestries. And using only those segments, we inferred what is called an admixture graph. And so using that, what you can do is now you can try to understand if assuming that each of these regions are independent populations, like sort of what kind of relationship have they had to each other in their history. And so what we see from this approach is a picture showing the peopling of Mexico from north to south, which goes hand in hand with what has been shown and considered already for the Americas as a large. And so the north of Mexico splitting off first, followed by the common ancestor with the common ancestor of all other regions. And then you have the split off of the common ancestor of the north of Mesoamerica and the occident of Mexico with the common ancestor of all the regions in the south and southeast. And that then splits off into the common ancestor of the center of Mexico and the Mayan region and the Gulf of Mexico and Oaxaca. So here, this relationship between the center of Mexico and the Mayan region was previously also, I was pointing out with the overlap we saw with the PCA and UMAP, and has been also brought up before in IVD analysis and Moreno Estrada et al. 2014, and is sort of pointing at evidence for an Atlantic coastal corridor of gene flow between the Yucatan Peninsula and the central Gulf of Mexico through all these different lines of evidence. So another thing that we can do now with these tracks of genomic segments with indigenous origins is we again can use them to create a PCA space that is only for sort of those indigenous ancestries, right? So we used here an approach called mass MDS. And first, what you do is created a space in mass MDS with these indigenous individuals self-identified from these different groups that are here labeled with text from a previous study, which is the Moreno Estrada et al. 2014. And on it, we projected just the indigenous segments of the Mexican individuals, which are shown here, circled. And so you can see that there is, those indigenous segments do fall often onto the groups that have sort of been common in those regions. But the main reason to do this, they want to show that, is to get an axis of ancestry within the indigenous ancestries, right? So this is thinking back to this tree, sort of we got through admixture, we quantified sort of this continental ancestry size, which has often been of a lot of interest, but we also, with the mass MDS on only indigenous individuals and segments, got an axis of ancestry within what we may call indigenous ancestries, and both will be useful in thinking later about complex trait variation, or at least we want to look at the impact of both. And so this was all sort of a picture of the ancestries in the Mexican Biobank, and the reason it interests us is because these quantities that we defined reflect these demographic histories that can be relevant for trait variation and complex trait variation. So to think about Mexico, first we can get a quick picture of what we know about demographic histories sort of globally, this is as learned from genomics. And so the key thing to keep in mind is the bottleneck that all individuals went through as they moved out of Africa and the peopling of Eurasia. And the second thing to keep in mind is the bottleneck that occurred here in the movement of people across Beringia in terms of populating the Americas. And this is most relevant for this particular study. So what we can do is we can use segments of the genome that are identical by descent. And so we call these throughout the Mexican Biobank, because we're interested in understanding sort of this later part beyond the Bering Strait bottleneck of demographic histories and what happened in terms of the peopling of the Americas and of Mexico in particular. We use these we call IVD segments in all our individuals, and we overlap them with these local ancestry tracks that were also called in all our individuals. And then we can use an approach called ancestry-specific IVD-NE, which what you do is you are inferring these effective population size histories over the last 300 generations within different within different sort of partitions of the genome to have origins of these different ancestries, right? And here we've colored these trajectories by what is known from Mesoamerican chronology. So in particular, you have the pre-classical period in blue, classical period here in pink, the post-classical period, and then the colonial and post-colonial period. So of course we can make out like there was a bottleneck in the colonial period, which makes sense. But there is basically what's happening here is that we're modeling together all indigenous ancestries, while as we had been looking at this, what that really refers to, the indigenous ancestries, is all the people who lived in Mexico at the time of the arrival of the Spaniards. And it's really a lot of different regions with a lot of different groups and indigenous peoples, right? So to try to get a better handle at those trajectories, which may be superimposed in that one plot, we repeated this analysis for these different regions. And so here, what you can see is, so this is the plot that was done for all individuals together. And then here, if we analyzed each of the different regions on their own. And so I think hopefully what's immediately apparent is that there's a lot of fine scale structure, like the different regions have different effective population size trajectories. And so to try to interpret that a little bit in terms of historical context, we can start with the south and southeast of Mexico, where there is historical evidence for large population sizes already. So, for example, in the Mayan region, already in the pre-classical period and in the classical period, a peak. And this was followed by what anthropologists and archaeologists understand as both a combination of ecological and political factors that led to a decline already before the arrival of the Spaniards. And this is what the genetic evidence is also presenting to us. In Oaxaca, similarly, there's there's archaeological and anthropological evidence for sedentary settlements already starting in the pre-classical period, followed by civilizations. So this is the Zapotec people, uh, and so on. Here in Oaxaca, which is followed by a more militaristic period and decline in population sizes. And so this is also what the genetic evidence is presenting to us here. So in the Gulf of Mexico, for example, among a myriad of other groups, we have the Olmecs in the pre-classical period, the Totonacs in the classical, and the Huastecs in the post-classical period. So for these regions, the genetic evidence also shows a peak NE already early on in the classical, and population style decline before the arrival of the Spaniards. And in particular for Oaxaca and Mayan region, it appears to show the population increasing even past the colonial period. And these are the regions with the largest indigenous populations and ancestries today, as also seen in the admixture. Now, this is in contrast to more the center and the north, where the Aztecs or the Mexica people had a stronghold, and these civilizations started later on. So it's mainly in the post-classical, and is also where the Spanish made their biggest stronghold. So in the genetic evidence is where we see the population rising in the post-classical and then falling, starting in the colonial period and continuing to in the post-colonial period. So obviously, showing this here, we had to make a association of generations to years. And so in general, from other studies, the range for humans is about 20 to 30 years for per generation. And so here I'm just showing the same analysis done for the two extremes. And more or less, sort of the results that I highlighted hold across this range. So we also did the same thing, of course, for ancestries from Western Europe found in the individuals of the Mexican Biobank, and similarly see that there is fine scale structure in the different regions, but in general that there are founder effects in all the regions, and the strongest founder effects for the Europeans coming to Mexico appear to be in the Oaxaca and the Mayan region. And for African ancestries, we also did this, and similarly also see founder effects and bottlenecks in all the different regions, and see this ancestry increasing in the post-colonial period in some regions, such as the Occident of Mexico, more so than in other regions. So this was all, getting some sense of the trajectories of these population sizes using genetics in the last 300 generations in Mexico. And of course, more work should be done on this as newer methods out, but for at least to us, this ASIBDNA seems the best approach so far to be able to both slice by ancestry and be able to recreate any affected population size trajectories using IVD. So we got a sense now of like thinking about genetic ancestries and how they reflect different demographic histories, both at the level of thinking about like, oh, indigenous European and African, but even within these, right? We saw how many different demographic histories exist within the indigenous ancestries concept or construct. So now these different demographic histories can basically lead to different patterns in the genome that are relevant for us, right? So for example, they can lead to different patterns of runs of homozygosity. So this is very similar to the concept of IBD in that if you have two parents with a genetic segment in IBD coming from a common ancestor A, then the child would have that segment in homozygosis or carry an ROH. So these can also be a window into demographic and mating history. So in particular, basically, if you have a large number of smaller ROHs can usually reflect bottlenecks or small population sizes held for a long time, whereas consanguinity or marriage among relatives will usually lead to long ROHs. And so there's a lot of work that has been done to show that, both through theoretical modeling as well as simulations. And so here we mainly learn from that and do an empirical study. And so here, what we did was call the number of runs of homozygosity per individual as well as the total length of the genome in runs of homozygosity. And in both cases, what you can see is that there is file scale structure there's more ROHs as we're moving south and southeastward in the country. So also the access along with indigenous ancestries are increasing. So to get a better understanding of that, what we did was we broke up the ROHs by size. And so here we have, well, first we just looked at it by ancestries, right? To be able to get at understanding if this is a relationship actually with ancestries. And so here we're showing the number of RH segments carried by an individual correlated with the ancestries from these different regions. This was quantified using admixture at k equals 5. And so clearly there's a strong correlation that there's more ROHs in individuals that have more of their genome with an indigenous origin. And then the rest of the ancestries show a negative correlation. And so to understand that a little better, and if that's coming from consanguinity or small population sizes, we broke down the ROHs into different buckets of size, and this was based on previous theoretical work from Harold and John Novembre, where a lot of modeling was done to show that this size bucket mainly reflects small population sizes or founder effects, whereas larger than this is often reflecting consanguinity in the population. And so what we can see here is that most of the signal that we were seeing is mainly coming from small ROHs. And so we inferred that to mean that the fact that we see more ROHs in individuals with more indigenous ancestries likely reflects these small population sizes and bottleneck and founder effects. So we also did this at the level of local ancestries along the genome and similarly showed that there's more ROHs per MB in segments of the genome with indigenous origins compared to with European origins, for example. And so coming back to this, runs of homozygosity is one way the genome can be structured because of these different histories. And so that's something we just looked at. But it can also lead to, for example, variation in the allele frequency distribution, right? So we had thought about these two bottlenecks. And so basically, again, a lot of theoretical and simulation work has been done to show that if a population goes through a bottlenecking event, you will have a change in the allele frequency distribution. So for example, here, if the balls are showing alleles, if you have rare alleles like the one in yellow, so some rare alleles will just be lost, like the one in yellow, whereas others might increase in frequency. So we want to try to see what's happening with respect to this. And given that we have just array data, what we were able to do is the following. So we count up the number of derived alleles carried by an individual. And we divide them into different functional categories, such as alleles that are intergenic versus coding synonymous versus deleterious. And then we again just do correlation with the ancestries in that individual from these different regions of the world. And we do this for all derived alleles that are rare, so up to a derived allele frequency of 5%. And what we see in this case is that, again, individuals with more ancestry from the Americas have a lower burden of rare mutations. And so this would be explained by if the rare mutations, because of their increased bottlenecking history, have been lost or have gone up to higher frequencies. So they have a lower burden. And so that's only for rare alleles. When we do this for all frequencies, there's basically no difference in the different ancestries with respect to the mutation burden. And this is in line with previous work also showing that even within Africans and Europeans, in the total mutation burden, there's no significant difference. But what's interesting is that it can shift the distribution, right? So there's fewer rare alleles, for example, in this case. And this is just a technical thing. Since we're working with array data, we did just correlate the burden that we got for rare alleles using the whole genome sequences that we had that we generated for 50 individuals from the MEXP, and showed a pretty good correlation between the two. And we repeated this whole analysis in the 1,000 genomes Mexican individuals and showed that this signal that we're seeing between ancestries and rare burden holds in whole genome sequencing data as well. So basically, this goes into thinking about the ancestries, reflecting demographic histories, creating patterns in our genomes. And so genetic ancestries don't only reflect demographic histories, but also environmental histories, and what we're interested in how these, and they also reflect different distributions of causal variants, which can be captured by polygenic scores. We're interested in how these can sort of create variation in complex trait and disease. And we also keep in mind that there's, of course, an environmental present, which can be things like experience of discrimination or different socioeconomic opportunities and so on, which while can and be independent of genetic ancestries in a post-colonial context such as Mexico, is often correlated and should be considered simultaneously, and that there are also other variables that we have just not captured in this study. So we're interested, and this, while not meant to be a comprehensive picture, is meant to be a schematic that we use to sort of get a handle at genetic and environmental factors and how they may work together to create variation, for example, in why some people are taller than others or have higher cholesterol and so on, right? So previous work, for example, have shown that ROHs, for example, have been associated with a number of complex traits. And so we work within this framework and the MEXP to think about this. And so starting with the classic complex trait of height, first we just looked at its distribution geographically across the country. And here you can see, so this is the empirical graph, that height on average increases as we move northwards in the country. And so to try to understand this pattern and what factors are leading to it, what we do is we model the trait distribution using a set of predictors. And so these include predictors computed from genetic data. So for example, genetic ancestries. And here, thinking back to our tree, we use both quantification of these continental ancestries from the admixture analysis, as well as this axis of continuous variation within the indigenous ancestries. Then other predictors from genetic data that we are interested in learning the effect of are the runs of homozygosity and polygenic scores that we compute on our data in a couple of different ways. And so we are also interested in predictors from demographic and environmental data. So here we use age and sex, as well as a number of sociocultural variables that we have access to in the MEXP, such as education attainment, whether they speak an indigenous language, whether they live in an urban or rural environment, as well as biogeographical variables. And we use two random effects to capture the genetic relationship matrix, as well as locality to capture any unmodeled environmental variation. So the goal with this is to ask, like, which of these factors are most relevant for explaining the variance of a given trait, and sort of try to quantify that effect. So this for combined models, we applied this for all the quantitative traits that we have. for example, explains 66.6% of the variance for height, 30% for BMI, 44% for triglycerides, and so on. So if we look at what we get out of an analysis like this for height, this is what we see. And so to focus first on the genetic factors, since this is a DNA dialogue, we see that the polygenic score is significantly associated with height. Having more ancestries from Africa is significantly associated with a higher height. Having more of your genome in ROH significantly associated with this lower height. And in terms of ancestries, indigenous ancestries, having more indigenous ancestries associated with a lower height, as well as this axis of ancestry, axis of variation within indigenous ancestry that's capturing sort of, for example, how much the Huichols vary from the Mayans is also associated with varying height along sort of a north to southeast axis. And so in that, there's also several environmental variables that are significantly associated in the joint model, but that genetic factors remain significant as well. And if we look at height over the years of birth that we had sampled in our data set, here individuals are broken down by the quantiles of ancestries that they have, indigenous ancestry. So what we can see here, the darker red are individuals with more indigenous ancestries do in our data set have a lower height on general, sort of going hand in hand with what we found from our previous analysis. But for all quantiles of ancestry across this year of birth that we're looking at are increasing in height. So this just shows the height increasing changes in environmental factors that happened over this period, as well as height reducing aging effects, both of which are insensitive to ancestry. So this picture is just to sort of show and remind again that even when we get from this like ancestries being significant, or it's not deterministic, and there's always environmental factors working as well for the final trait value. So we did this for a number of different traits that are all available through the manuscript, but just to give a few examples, here for glucose, as well as triglycerides as well, indigenous ancestries were associated with a higher value, whereas ROHs are associated with the lower levels of glucose, triglycerides, height, as well as BMI. In particular for BMI, where obesity is often talked about as a major problem in Mexico, we don't see any significant association with ancestry in the full model. But if anything, we see ROHs being associated with lower BMI values, and ROHs, as we saw before, are enriched in indigenous ancestries. So here now, if we look at the different genetic factors that we looked at, which are these different ancestries, as well as ROHs and PSs, we can see a summary of basically all the traits that we looked at and what they ended up being significant for. So ROHs ended up in general being associated with lower values of traits and was significant for high glucose, triglycerides, and BMI, and so on and so forth. Polygenic scores being significantly associated with all traits. We can also look at the summary for the environmental variables, which also end up being, for example, living in an urban environment is significantly associated in the full model, jointly modeling genetic traits with higher LDLs, HDL cholesterol, creatinine levels, higher BMI, and higher height. So all of this was, I felt it important for any future work, including our own, to make these sort of trait profiles to first understand, epidemiologically speaking, what are the different factors that are involved, and we think this should be relevant for both future GWASs as well as public health interventions. But to go down sort of what's more standardly done with genetic studies, what I'll end with is we did to look further, understand the genetic architecture of these complex traits. We did do a GWAS for all 22 binary and complex and quantitative traits that we have, and also try to get a handle on polygenic prediction. So this is just a picture of all the traits that we have. And so what we did for the polygenic prediction aspect is we broke down the data set into two sets. And so we did a GWAS using Regeni in about 2/3, and then we created polygenic scores in the remaining 1/3 using our own GWAS and compared that to if we use summary statistics from the UK Biobank GWAS to look at prediction performance. So for example, if we look at the results that we got, even at 6,000 individuals, we were able to validate genetic associations with cholesterol and triglycerides, all of which have been previously found in other cohorts. So when we did the GWAS with Regeni, which was the most conservative approach you could think of, we didn't come upon any new associations, but did validate several that have been found in other cohorts, including the ABCA1, which is an allele that's basically predominantly found in the Americas and a little bit in East Asia, and does not really have a frequency in any other part of the world. So using this and making the polygenic scores as I described, which we can do in both the genotype data set as well as the imputed data set that we imputed using TopMed, I think the main thing to see here is that even with the smaller size of the Mexico Biobank, this first pass, which is 6,000 compared to the UK Biobank, which is like 350,000 individuals, we see a better prediction performance using the MEXP GWAS compared to the UK GWAS for triglycerides. We see the same thing for glucose. Whereas for some traits such as BMI, the best prediction is seen using the UK Biobank GWAS as well as for height. So just to give some examples. And while there's likely empirical factors at play, it's probably also theoretical factors of why different traits have different transferability across GWAS. So this can be the mutational target size of the trait and the heritability per mutation, as well as the strength of selection acting on the trait. So traits like BMI and height, which are highly polygenic, it seems that the best powered GWAS gives the best prediction, even if it's from a different cohort versus traits with likely a smaller mutational target size, such as triglycerides and glucose. It seems most relevant to just have a cohort that's more similar. So we're using our MEXP-based prediction. Despite the lower end, we got better prediction for triglycerides, glucose, creatinine, and cholesterol levels. And so this is the ABCA1 gene that I mentioned, which, as you can see here, is predominantly found across the Americas, but not really elsewhere in the world. And I think I'll just point out that while we find it significant in our GWAS as well, and while individuals who have larger ancestries from the Americas do have a greater frequency of this allele, and the ones that have a higher frequency of these alleles do show a lower HDL level in our data set. Nevertheless, in our joint model, when we look at these indigenous ancestries as a whole, ancestries from the Americas are not associated with HDL, right? Whereas if anything, ancestries from the Americas are associated with lower HDL levels, which is actually a healthier profile. So these results illustrate how cultural diet factors and genetic factors are essential for different cholesterol aspects, but also imply that while some functional variants may be specific to regions or genetic backgrounds, these are few, and cautions against using global ancestry proportion as a predictor of the effect of single functional variants. So the perspectives that I really found while working on this that I would leave with is that I'm thinking about complex trait variation. We have to embrace complexity from genetic and environmental sources and the interaction. So a lot of work that I'm focusing on now, both with the MEXP and the MCPS, which is a large cohort from Mexico City, is now thinking about the G by E, for example, with respect to BMI. And that ancestries must always be considered at various timescales to avoid oversimplifying and essentializing different groups. So in conclusion, we see multiple genetic ancestries in contemporary Mexicans, diverse in origins and variable across the country in line with historical evidence. We show elaborate fine cell structure and population histories as inferred through IVD and local ancestries in different Mesoamerican regions. We show the large number of short ROHs within indigenous ancestries due to these multiple bottlenecks in small historical population sizes. And that as a result of these, we also see lower rare variant burden within these indigenous ancestries. And that while ancestries and ROHs are an important predictor for some, but not all traits, and can be in different directions. And the polygenic prediction is better using the Mexican Biobank GWAS for some traits, despite the smaller sample size at the moment. So this study is publicly accessible and is deposited in the EGA. So we both have 50 whole genomes as well as mega-array data for 6,000 individuals, and all information is in the manuscript. So this was obviously a huge team effort. And so in particular, I'd like to thank Andres Moreno Estrada from Langebio and Lourdes Garcia from National Institute of Public Health of Mexico for giving early direction to the project and winning the funding for this first phase, as well as John Novembre, who gave a lot of conceptual input for the population genetics and statistical genetics analyses. And these are the joint second authors of the paper, who helped lead both generation, PopGen, and the GWAS analysis. So recently we put out this perspective, which gives more insights into just the back end of what it took to generate this data and building the local capacity to pull this off in Mexico. And so since a lot of the focus of this project has been, beyond also just the genetic work, to build local capacity to do this work and make it self-sustainable in Mexico. So a lot of students now are working on new projects using this data set. So these are students in my lab, they're students in Andres's lab. And so this is ongoing work using this data set. And I was thinking to talk a bit about, but I think I'm out of time. So I'll just thank my lab at the Center for Genomic Sciences, and just place in context where the Mexican Biobank comes in. So the reason why it says 40,000 here is because we do have 40,000 individuals collected that can be genotyped and sequenced for next phases of the project. And this is just to show sort of the increasing biobanks in the global south, as well as compared to in the global north, where a lot have been prevalent now for some time. And that, thank you all for your time, and thank all my funding sources, and happy to take questions.
Mashaal, thank you so much. What a fascinating presentation. And, thank you for, the way you communicated your work, made it very accessible for those of us who don't have, the expertise in sort of the population genomics, and that particularly the sort of structural and the ancestral work. We have got a few questions. So, I think to start off, it was very interesting that Mayan region standed out quite significantly from the UMAP analysis and other analyses. Forgive me if I missed it. Did you do the ROH analysis regionally as well? I know it was sort of a proxy for indigeneity, but did you do that analysis regionally as well?
So the ROH were called on each individual, and then we looked at them a few different ways. So we looked at them sort of putting the individuals in buckets of states. So it showed that, and that showed the ROHs are higher in states with more indigenous ancestry. So
okay, in the south
and
and
southeast.
Yeah, okay, got it.
Yeah. And then we did a correlation of the ROHs with the quantified ancestry from each region for the individual. So we looked at it with respect to the proportion of their genome with European or indigenous or African ancestry.
Okay, excellent.
But they were called by individual. Yeah.
Yeah, got it. All right, we have a question from Nicholas. Thank you for the fascinating talk. How has the genotyping platform influenced or limited your analyses? So particularly with the ROH work, are you not capturing indigenous ancestry variation in the genotyping that breaks up the ROH?
So that's a great question. So the genotyping platform that was used for this was done after a lot of thinking. So it's a mega array, which it was designed. So it's designed not by me, but people like Andres and others who have previously been involved in creating more genomic resources for Latin America to exactly ensure better representation. So it actually captures quite much better than other arrays, just indigenous ancestries or variation. And so one of the things we saw, for example, is captures rare variants quite well as well if we compared it to the whole genomes that we have. So while it's still a limitation compared to using whole genome sequences, obviously, it's actually an array that was designed to capture well sort of global variation.
Okay, understood. Michael asked a question that I was also interested in. So what was the most surprising challenge or barrier that you faced in establishing the biobank? And I know that you were working on the biobank samples and didn't or weren't directly involved in sort of establishing the methodology for recruitment, etc. But we do think a lot about strategies for recruitment in different regions into genomic studies. What's the general sort of public acceptability or relationship with enrolment and participation in biobanks and studies like this? Was there any regional variation in a strategy to recruit members of the public?
Mm-hmm. Yeah, so that's also a great question. So the way that it worked out here, and I think is generally a great strategy to take, is partnering with an institution that is sort of well set up to do that kind of things. In this case, we partnered with the National Institute of Public Health that is set up and sort of periodically does these surveys across the country and have the personnel who are trained in how to do it in a culturally sensitive way. So they set up engagements depending on if it's in the rural groups of the community leaders or if it's in urban areas, depending on where the sampling is done, to explain, like all the ways that the data could be used in terms of for health studies and so on. And then they do like household visits. So in this particular study, there was household visits of like a couple of hours in which they do these long questionnaires with the individuals. So that's, and we go into more detail about this in the perspective piece that I pointed out. So we basically partnered and in that sense outsourced all that part to the National Institute of Public Health, which has been like sort of set up to do that. And in that sense, they had all the personnel and the training and the engagement to do the recruiting. And we came in at the part of then using it for genotyping. So in general, I think sort of reinventing the wheel, it's like a great strategy to find and work with institutions that have been working at that level at the field across the country with groups at different in rural urbanities and urban localities as well, and sort of use that expertise that they already have. So that's the approach that we took here.
Okay, thank you. Regan asked if there was any communication about results back to participants of, who had contributed their data and samples, and sort of also about the broader challenges. What were some of the strategies or challenges faced in this process?
Mm-hmm. That's also a great question. So the Mexico Biobank, the way that it's structured compared to, like, for example, the previous study that I talked about, which is the NMDP. So the previous study went into self-identified indigenous groups and sampled from them and did went back and gave the results to communities. The Mexico Biobank, and then is sort of set up as a national scale, and so in that sense took samples from many different regions, and no one as such like self-identified. We don't even have that information. It's just that we're covering rural and urban localities, and in that sense cover a range of different ethnic, ancestries and so on. So in that sense, the way the communication has been done as well, so we don't have recall, right? Like we don't have addresses to go back to, is to do it also on that national scale. So all the so I have done a lot of it, and so has Andres, is to do it through outlets such as television, newspaper, radio, museum talks, making infographics. So that's the way that we have done it, and both explaining so try and explain very carefully, like the way I did now as well, that ancestries is on many different scales and environmental factors, as our own analysis are showing, are very important for things that maybe have been attributed to ancestry, such as obesity and so on. But so that's the level at which we're doing the communication, sort of broadcasting it using these platforms, since that's also the way the data was captured sort of across the nation, and we don't have recall addresses or anything for any of the samples.
Okay, understood. I did note that with interest when you said, BMI is perceived to be a regional problem in Mexico. However, it was sort of a low association with the Indigenous ancestry. On the basis of that, can you see this body of work informing health policy? So health interventions for different regions or populations. Are you engaging with sort of health policy makers, I guess, to inform them of your work and implications to their strategies?
I think 100%, like, and that's sort of something we talked about, at least like lightly in the things that it seems like if anything, there might be like G-by interaction, or that environment is very important for obesity, but at least in a joint model, just ancestry is not relevant the way it shows up as relevant even for something like height, right? So it's definitely not just that indigenous ancestries are doomed to be more obese or genic or something, right? It definitely seems either because of diet and so on, because also living in an urban area is significantly associated with higher BMI, or it's some interaction going on. So that's why in a project that I'm developing now, we're going to really carefully try to assess like if there's G/E that's going on for BMI in particular. I think that really informs a direction where the country is already going in terms of really putting attention on diet and making public campaigns to, make people aware of that.
Fascinating. Mashaal, we're going to have to wind up, but thank you again very much for your presentation. It was really fascinating work, and all the very best with the work as it progresses.
Great. Thank you so much. It was really great to be here. Thank you.
Thank you. And thank you all for joining us today for this presentation. Please join me for the next DNA Dialogue webinar on Thursday, the 28th of March, when I'll be joined by the lovely Mark Fumé from DNA Stack. He'll be presenting a presentation entitled "The Future is Federated, Accelerating Collaborative Research with Standards-Compliant Federated Networks." I hope you'll be able to join us. And Mashaal, thank you again.